Thank you very much, Mon. Thank you very much, Andrew, for the great invitation. It's a real pleasure, one of the great honors of my academic life, I guess, to be invited to be giving this plenary talk here in the city, which I love so much. It's not the first time I've been here. I think there's the third academic primary care conference that I've attended in Galway. And one of them was, I think, the Society of Academic Primary Care that Andrew organized. And on that occasion, Andrew organized a sort of, it was a four-nation football tournament. And I was playing for Wales. And I got a feeling we got into the final against Ireland. But yeah, Andrew brought on some ringers, his actually sons, who were really good, a lot younger with fresh legs. And I think it was a wipeout. It was a bit... They played like the current Irish rugby team, let's just say. So it's a great pleasure to be back, and this time to be talking about, I guess, changes in the design and delivery of clinical trials. And I'm focusing on acute therapeutics here in primary care, and we'll be looking really at antivirals. But the principles of doing this kind of trial, I think, does carry over into trials of chronic diseases, or in fact, you know, there's some sort of basic issues that we need to get right in order to do things better. So just to go back to, sort of a lot of fresh faces here, but most of you probably are old enough, certainly Tom, myself, will remember the swine flu pandemic. You know, not a single person actually was randomized in the community in a trial during that pandemic. And we gave out this drug, oseltamivir, in large quantities. And it still remains one of the most contested drugs on the planet, actually, because people, although it's in guidelines, people sort of don't use it. People sort of don't believe the evidence. That could have been solved if we had randomized people during that time instead of just telling people to take it if they had the syndrome. Because we know we did a lot of harm. We gave drugs to people without seeing them, certainly in the UK. And some people didn't have acute viral illness, but pneumonia, meningitis, and came to harm as a consequence of that strategy. And we don't know how much good we did or how the balance was. So there's still, you know, lots of systematic reviews about that drug for seasonal flu, and you know, we still see comments like we don't really know in who we should be using it and when. Problem arises, I think, that when you know we start with a pandemic, we get a quick public health response. In my own practice in the South Wales Valleys at that point, a public health physician helpfully said, every time somebody gets a cold or upper respiratory infection, you've got to put on your PPE and get a mask and go out there to their house and swab them. Don't bring them down to the practice. Now, I don't know if you can imagine how many people have coughs and colds, and if the GP's got to do that and go to their house, not bring them down to practice, it's very unhelpful. But nevertheless, the public health response was quick. And by the time you get to preclinical research, the clinical response, the pandemic is usually done and dusted. So what we're aiming to do is this, to bring forward the clinical research so that we can start trials early on in the pandemics or outbreaks, or if there's interventions to be evaluated even in epidemic situations much earlier on, so we can generate evidence that will help care, that will underpin care during the pandemic itself. And I think that's the goal. So as part of this European Union-funded project Prepare, and several colleagues in this room were involved in making that trial happen, we did what you might call a peacetime trial of over 3 winter seasons of looking at this drug, oseltamivir, for influenza-like illness. And you can criticize this research in many ways, but interestingly, the drug company trials done by Roche, there are about 20 of them, they were systematically reviewed in many different ways by many different bodies, and they all came up with the same conclusion. Although the teams that were doing these systematic reviews didn't get on really and criticized each other's methods, the results were all the same, that it's going to help you by a day, give or take an hour or two. So with this ALICE trial, we recruited 3,200 patients, and we found exactly the same thing as the placebo-controlled trials that Roche had done, that on average you're going to get better by a day quicker if you take this drug, if you've got flu, both laboratory test proven or the syndrome. But what this trial added, and we did this as an open trial, so it was not placebo-controlled trial, and that's interesting that it gave you the same answer as the placebo-controlled trial. But we used a platform trial design that allowed us to identify certain subgroups that might do better. So the estimate, for example, for older people who were sicker, who had comorbidities, they would get better by about 3 days, actually. And so on average, we were able to say, yes, you're quite right. There's not much point in treating an otherwise healthy person who gets the flu with this drug, but it could be beneficial in certain subgroups. And so designing a trial in that way to give you power to estimate subgroup benefit was an important addition, and we used that using Bayesian statistics, where we did a lot of simulations to power for each subgroup. So I think that brought us a little bit closer, that experience brought us a little bit closer to the vision of randomizing patients within a couple of weeks of the onset of a pandemic that could help underpin care during the time that people were so sick. Now, while the COVID pandemic has been going on, and indeed the swine flu, there's another pandemic going on, and that's the pandemic, shall we say, of antimicrobial resistance. And you know, we found a lot of hand-me-down evidence from hospitals to primary care, but the intended use population is different. And you can't assume that evidence from one place is going to apply to another place. And the whole mission here, I think, is to try and target drugs in general, and this applies to drugs for chronic conditions as well, to those who are going to benefit and keep them away from those who don't. And you know, one of the big problems we face in healthcare is that we're giving people lots of treatments that actually have got no chance of benefiting them. And I noticed this when I came to the UK from South Africa. You know, the same antibiotic that we were giving to people who were virtually dead, and 5 days later they would get up and walk out the hospital, we're giving to snotty-nosed kids. And you know, the question came to my mind as to why we do that. A very complex solution to that, but getting the drug to those who are going to benefit, keeping them away from those who won't. And this issue is really, I suppose, what I think of as having respect for medicines, particularly antimicrobials, because we do a lot of harm when we use them unnecessarily, going back to that pandemic of antimicrobial resistance. And this blue plaque sits on the wall outside the Department of Primary Care at the University of Oxford, which is the old Radcliffe Infirmary, where penicillin was given to the first human being. And the other side is actually the clinical notes of that first human being, Mr. Anderson, who was a policeman. And they are in the Bodleian Library that you can see. And it's actually quite a moving thing if you were to read those notes verbatim, because he has a story of a man with a sort of an abscess on the side of his face. It's unclear whether he was in Southampton apparently during the Blitz, and if he got injured helping after a bombing raid, or some people say he got scratched with a rose. But the etiology of it is not reflected in the notes. But what is reflected here is that he was really going downhill, and people came from the Dunn School of Pathology with some penicillin, and they gave it to him. And you know, what I witnessed in rural South Africa was happening now to this man. He was taking up his bed and ready to walk until they ran out of the penicillin. And these notes reflect how then he deteriorated, but they were purifying his urine. And they got that penicillin back from his urine, they gave it to him again, he started to improve, they ran out again, and he rapidly deteriorated and died. And I show this because actually Fleming predicted that the misuse of penicillin would lead to antimicrobial resistance and would lose the power of the drugs. So that is also true of antiviral agents. I think we can't make assumptions that treating the wrong people is a trouble-free thing. Now, we've based a lot of what we've done with antibiotics on what you call it assumption-based medicine. We found that it was good for that man, Mr. Anderson's, good for people in the ICU with pneumonia, and therefore it's got to be good for people who are coughing, and therefore it's got to be good for a kid with an earache or whatever. But the problem is we never did the clinical trials in those populations. We ended up using it without knowing if there was a real benefit. So it was an assumption-based medicine. And interestingly, I think there's a lot of, problems, let me just say, about the way we focus on the tip of this iceberg, the top of the pyramid. So the COVID pandemic, for example, the recovery trial randomized a single patient, and the chief investigator is standing on the television next to Boris Johnson. And, the press seemed to love the ICU bringing people back from the jaws of death. But in a sense, it's in the community where early treatment perhaps has got its greatest potential, reach and impact, because if you can stop people from getting there to that position where they're facing the ventilator, that's going to be much better for everybody. But that's not where the funding initially goes, and that's not where the media interest is, or the political interest. And that's a big challenge, I think. Overall, less than 20% of clinical trials around COVID were done in the community, and most of those produced no useful information. There was a lack of collaboration across countries, between countries, and within countries, and I think something like 120 trials at one point going on for one drug, hydroxychloroquine. And the trouble was that people were kind of nationalistic, they weren't collaborative, and they couldn't all recruit, and the regulation was a nightmare. And that arose from the fact that we have no standing infrastructure for primary care clinical trials across Europe or across the world, if you like, that could do what I was saying earlier on, randomize people quickly to get the evidence that we needed. So we started in the UK. We had some funding from the UKRI available, and I led an application for that. And the point was to combat this many single drug trials that weren't recruiting, or that could never recruit to target. And we needed to rapidly initiate a trial that had adaptive features, because there was hydroxychloroquine, everybody wanted to do that drug, but we knew that there would be others. And starting a trial every time there's a new drug is a wasteful thing, because that takes setup and costs and so on. So a platform trial where you could add in new drugs as they became available or were discovered would be much more efficient. And we knew that we couldn't do a trial that, shall we say, lived up to the expectations of the FDA or something like that. We were trying to answer a different question to the placebo-controlled trial question. So I'm not saying there isn't a place for this. In efficacy trials, you need placebo-controlled trials, no doubt about it. But that kind of trial says, if I give this drug, is it going to be better than giving the drug or sugar-coated or dummy version of the drug? That wasn't the idea here. Our idea was to say, if I as a GP, say, prescribe this drug, am I doing more good than not prescribing the drug? So the control condition that we decided on, also for logistic reasons, was not prescribing the drug, usual care without the drug, if you like. Now, a lot of people don't like that. A lot of people will say, if it's not placebo-controlled trial, it's pointless. Nevertheless, that was the design that we came up with and was funded. And looking back on it in 2020, I got the signal to get on with it on the 11th of March. I had COVID at that point myself, had the first draft of the protocol based on the Alice study. We couldn't have done it without that European Union funding and those relationships and statistical input and knowledge based on that. And we got ethical approval within a week, and so it can happen, and got our first patient in on the 2nd of April. Now, we didn't recruit to the same extent, for example, as the recovery trial did, and there are practical problems for us in general practice. Like in the UK, there's over 9,000 practices, and you can't set them up as clinical sites. We set up about 1,000 within a matter of weeks, and that was quite hard because they need contracts and GCP training, and you name it, you know all about it. And not all of them can be, a lot of sickness, and not of them are not interested in research or can be involved, and none of them have a pharmacy that can label medicine and give it to a patient who's essentially a captive audience. We got bits of hydroxychloroquine, in fact, in packages of the wrong size packages, and the GPs had to cut this with the scissors, repackage it, relabel it, and to get permission for people to relabel this drug was very difficult. In fact, somehow the MHRA, the regulator in the UK, gave me permission as a single individual to start relabeling the stuff. So I sat there in the trials unit, sticking labels onto boxes through the night, and then posting. So the regulation, the point of that story is the regulation was not fit for purpose. And that's a piece of work that we've still got to do. And no sooner had we set up these 1,000 general practices that the government said, if you've got COVID or you think you've got it, don't go and see your GP. Phone NHS 111. So I thought, oh, great, well, they will recruit from there. But NHS 111 is the most fragmented organization, or it was, that you can imagine. And I don't know how many meetings we had with them, but they couldn't adapt. And they couldn't get the permissions, and the research prevention police were all over this, data protection, you can't introduce a trial, you can't give their information to us, etc. So that kind of never worked. Spectacular failure, but there we are. So then that kind of takes me back to March 2020. That's how I was feeling, I think. But this is what the principal trial was. It was an open label trial. It was a platform trial with an overreaching master protocol where you could add in intervention-specific appendices as you got new drugs into the trial. And they were major amendments, but you didn't have to start the trial from the get-go. We did, for those with a statistical interest, we built in the capacity for response adaptive randomization. And this is an interesting thing whereby you can alter the proportion of patients that are randomized to different arms as the trial goes along, so that a trial arm that's doing better can get more people allocated to that trial arm, and theoretically that'll give you an answer quicker. And it has the advantage also of meaning that more people have a chance of getting a better performing intervention during the course of a trial. We also then kind of had to innovate, because NHS 111 and the practices at that point weren't able to help us with the recruiting. So we started to centralize recruitment and open up ways in which people could participate in the trial without ever leaving their homes. And in that sense, I think this became the first truly democratic, nationally inclusive trial where we would take the research to where the patient is. Because traditionally in general practice, we set up a site, and a patient can come to that site and be part of that research if that's where they're registered. Well, that's how it is in the UK. That means that those people who aren't registered at a practice that isn't doing the trial, that is doing the trial, they can't be part of the research. So it limits people's joining research by virtue of where they live or where they receive their health care. So we want to make a sort of low-burden trial that could, for the patients as well as the GPs. And, you know, I'm sure Tom might even have known Julian Tudor-Hart, but for those of us who sort of enjoy the sort of background philosophy, shall we say, of general practice, is a great doyen of GP. And he said the availability of good medical care in the population varies inversely with that need for that medical care. And he said, incidentally, that this inverse care law contrasts, it operates more completely where medical care is most exposed to market forces. But nevertheless, we sort of thought about this, and there he is in his population, Glencorrig, where he got just about everybody included in some MRC-funded studies. But it kind of seemed to us that there was an inverse research participation law, which I think as primary care clinicians we need to, and researchers need to address. So I won't go into the sort of challenges of the two-arm fixed allocation trial, beloved of so many. The platform trial design is where you can have several drugs with different groups. So you can get, say, a person with characteristic A, you'll get an answer for perhaps these 4 drugs here, and different characteristics can come into it. So you can power your trial to get answers by different demographic features and for different drugs in the same trial. And that differs from what you might call the basket studies, where you're looking at a single trial, looking at multiple subgroups of interest. And similarly with the umbrella trial, you have a single person or single homogeneous population with many different types of drugs that characteristic is exposed to. And I think this explains it quite nicely. So the black line here represents the control group, and A, B, and C are the drugs, of course. And the trial starts. Now, this is the beauty of it. You don't have to start a new trial for drug D and E. And because you do regular interim analyses, you can see that early on that you've got a drug C here, that is causing harm or is futile. The prior probability that, the pre-specified probability that this drug could be beneficial is so low that you can drop it. You can then combine drugs if you think, well, particularly important, say, with antivirals. You might think, actually, just giving one antiviral could be generating resistance here, and you need a combination. We find here that drug B is superior, and that can then become part of the standard of care. So you're not sort of limiting people's ability to benefit from drugs that are proven to be beneficial, and in the course of the pandemic that has happened. So this is what we did with this principal trial. We had 7 drugs under study, and I think we're now just doing the analysis. It's taken us a long time to do the analysis for ivermectin and favipiravir. But I think very early on we got evidence that doxycycline is an antibiotic that was thought to have anti-inflammatory properties and antiviral properties, as well as azithromycin. We showed that those didn't work, and I think that was part of a therapeutics alert. The beauty of the NHS is that its chief medical officer could tell every doctor the next day not to use these drugs, and that was, I think, a bit of a home run for the mission of antimicrobial stewardship. Now, based on that trial, we got the second trial funded to address the issue of novel antivirals that were being invented, and you might know of the drug molnupiravir and Paxlovid, and they're currently in this trial. And while principal focused on the repurposed medicines, this panoramic is focused on the novel antiviral drugs. And we also wanted to include virology sub-study and doing a health economics component. And a lot of people criticized us and said, look, you're killing people by doing this trial, waiting for the trial results. Because there is a trial, it was called a move-out trial, which shows this drug works. Except that that trial was done largely in the Russian Federation and in Brazil in unvaccinated people. And for those who've scrutinized it, there are questions. And a different variant was circulating at that time. And again, I go back to this issue of the intended use population. Did this drug work now in a population that was in developed country, predominantly in a multiply vaccinated population while Omicron was circulating? And so that principle comes down to it. People could be involved if they had an underlying health condition to the age of 50 and anybody over 50. And again, it was an open trial. And we opened actually on the 8th of December, and it was a day that UK Prime Minister, he was scheduled to announce this trial, but in the end he decided to focus on some other troubles that he had at that point. That was the day that Allegra Stratton resigned after discussing whether parties were parties or whatever. I won't say any more about that. So there were, we worked very well, I think, as a 4-nation study, and recruited through the multiple methods that I've already alluded to, and people could register for this trial online, get randomized, get a call from a clinician, discuss participation, go through the information leaflet, and then they would get couriered medication directly to their home, and follow-up could be online and so forth. And we did, on one day, randomized 540 people, which I think was the fastest recruiting trial of a medicine ever in the UK, if not anywhere. Not that we're competitive, but we recruited faster than the recovery trial in hospitals. And we were able to use a lot of digital enablement. I mean, there was about a third of the patients were recruited through general practice in this molnupiravir comparison. But we were able to actually text people who had had a positive test and to reach out to them to see if they were interested. And NHS Digital gave results back to all 4 nations, but through the England data, we managed to get some contact details, which ethically is an interesting thing that we can perhaps discuss, but bringing trials like this to the attention of people is important. So we finally randomized, we screened actually nearly 200,000 people, but randomized nearly 26,000 You can see the weeks of recruitment has gone down because the weekly recruitment, because we now are recruiting to the drug Paxlovid, which is much more complicated, and you need to see their medical record in full, more or less, and many more exclusions for that drug. So the molnupiravir, this novel antiviral from MSD, we found, well, in a nutshell, we managed through this mechanism to have a nicely balanced, the randomization worked, we had a high proportion of minority ethnic people. We had a particularly innovative, I think, outreach program. We employed Mahendra Patel, who is sort of expert in many contacts and in faith groups and professional groups, and we went and visited places of worship and community groups and professional groups, and the minority ethnic proportion in this sample represents more or less the same proportion in the general population for the age groups concerned. We managed to sort of randomize people within 2 days of the symptom onset, and over 90% said that they took the drug. Now, the primary outcome for this trial, the government really wanted to know, does this drug keep people out of hospital? And because hospitalization rates were so low, that is why we had to get the sample size so high, but actually it didn't. It made no difference to the hospitalization rate, but these were vaccinated people where hospitalization was very rare in any case. We looked closely, going back to what I was saying about the platform trials, whether there was any subgroup that could be seen to preferentially benefit from this drug, and actually there were none when it came to hospital admission. It was distributed evenly by any kind of way you wanted to cut it. However, this drug did make people get better quicker. The median time to recovery was 9 days amongst those who took it, and 15 days who didn't. And there were a lot of other secondary outcomes, such as sustained recovery. There's lots of bumps in the road when you're getting better from the COVID, and what we found here is that for those who got better, they stayed better more often, and they consulted their GP less often a little bit. But that benefit was equally distributed, so no subgroups really benefited. Now, we found also that there's a mechanism as to why people will self-report improvement, that fewer people will excrete the virus over 7 days, and that the viral load will be lower. But go back to my kind of thinking about penicillin and antimicrobial resistance. If you look here at day 14, these kind of dramatic differences disappear. And in fact, the viral load kind of goes up a little bit in those who took the molnupiravir. And I can't share the results with you now, but we're having a look at those strains now to see, firstly, are they infected? And molnupiravir works by making mutations in the virus. These are supposed to be catastrophic, and maybe they are, and the virus can't go on. But we're kind of looking at that because, using a drug at scale like this just takes one of them to start changing, and, well, as we know. So with this drug, it didn't reduce an already low hospital admission rate, but people got better quicker. There was less healthcare seeking. It had an impact on viral load and recovery over 7 days. So what should you do? I suppose, well, this drug might help people who might help in a situation, say in pandemic, where nurses are unable to go to work because they've got COVID, or prison warders or whatever. So you might want to use it in key groups, but if you're using it to keep people out of hospital at scale, don't. So what helped us achieve this? Well, I've mentioned this already. So the experience of the EU-funded trial, ALICE, that many colleagues in this room helped with, got us able to get going so quickly. The peer-reviewed funding that we could access, that was nimble, it didn't take forever. In the UK, they had a thing called urgent public health banding. So instead of having 200 trials, each a small one, they had national priority trials, the REMAP CAP trial in ICU, recovery in the hospitals, and principal and panoramic in primary care. And they kind of limited NHS resources to those trials. That was very helpful. We had a standing infrastructure, and I can't emphasize this enough, in the NHS, predominantly nurses helping with secondary care trials, but we were able to deploy the clinical research network to great effect in these trials, and that was amazing. Eventually, we got an independent panel which reviewed applications for drugs to go into those trials. Because, obviously I don't know enough about, it's not up to me. It shouldn't be up to the investigators of a platform trial to decide which drugs they're going to pick. You need all the basic scientists as well as the epidemiologists saying that's a good bet. Then we got the digital data for follow-up after a lot of hassle. Nigel will attest to that. We did heroic work, certainly in all 4 UK nations. And then, that online consent and follow-up and that central distribution was important. So my sea change, there's Ariel's song. I don't know what this does for the traditional way of doing clinical trials, certainly for acute therapeutics in infectious diseases, but Ariel's song about a sea change is about this body that is now being transformed into something that will last and be beautiful, and I hope that that's the case. So there's still ongoing challenges, lots of skills shortages, design, delivery, and with that 7, I showed you where we've had 9 drugs under study during this time, and at that point, when one drug is coming off the production line, as you say, you're introducing a new intervention-specific appendix, you're trying to get a paper published while you're trying to do other stuff. So it's like doing 9 trials all at once. The big problems with the data protection and kind of defeats the patient autonomy. When the patient says, yes, you can use all of my health-related data, and then when you say to the people who own the data, here's the consent form, can I have the data? They say no. There's so many different regulations. We try to get these trials up and running in many different jurisdictions, and there is versions going in Canada now, but getting the standing infrastructure with all the regulations and permissions in advance is so important before trouble starts. So I think just, these nurses who put up their own blue plaque in our trials unit, but they, the team kind of worked their socks off. And just to say, people might be thinking, what's all the fuss about? This COVID's all gone and stuff. Except that I don't want to alarm anybody, but this bird flu has killed millions of birds, and it's now in mammals. And we can't wait for it to jump before we set up the structures to do the research, which is so critical to pandemic preparedness. So thanks to everybody, and thanks for listening. Sorry, I've spoken too much.